everyone. Our today's topic is anatomy and morphology of plants. The learning outcome we are going to accomplish are: first, to understand the anatomy and morphology of plants; second, to differentiate monocot and dicot plants based on their physical feature. Monocots versus dicots. First, we are going to talk about the root morphology. You know morphology, right? Morphology is what you can see from the outside, how a creature appears. So first, we are going to talk about the root's function. The root's functions are: first, to absorb water and dissolve minerals in the water; second, to provide an anchor that keeps a plant in the soil; third, to provide a support; fourth, the root serves as storage organ for water and carbohydrates, as in the modified swollen roots of carrot and sweet potato. Next, aeration. Plants that grow in stagnant water or other watery places have modified roots called pneumatophores, to which oxygen from the air diffuses. Last but not least, another function of root is reproduction. The plant root system also serves as a natural means of perpetuating a species. You see on the screen now the kinds of roots. All roots are responsible for anchoring the plants to the ground, extracting water and minerals from the soil, and this is what we call the taproot, owned by dicots and gymnosperms, while this one is what we call the fibrous root, owned by monocots. And there is another one called the adventitious root, which is kind of additional kind of root. I mean, you see, this is a stem, okay, and from the stem came out the adventitious roots. This table on the screen shows us the differences between the three types of roots: taproots, fibrous roots, and adventitious roots. First, taproots. They are large and fleshy in appearance. They grow deep into the ground to search for food, and they are able to store food, which is particularly necessary for perennial plants during periods of dormancy. And taproots are mostly found in plants with two seed leaves, cotyledon, called dicots. Fibrous roots. They lie close to the surface of the soil. They collect precipitation before it sinks deep into the ground. They can often be found growing side by side with taproots. The two types of root combine efforts to maximize efficiency, and fibrous roots are mostly found in plants with one seed leaf, monocots. Next, adventitious roots develop to help the plant climb, for example, ivies. They help develop modified underground stems such as bulbs, and they are common in both dicots and monocots. Now, you also see another picture on the screen. It's the image of a root. This is the appearance morphology. You see there is an area on which you can find the root hair. We call it the root hair zone, and this is the elongation zone, the area in which cells are elongating. And then at the end we see also the root cap. Okay, root cap will protect the root tip, so that when it penetrates the soil, it will be protected against the friction or any mechanical damage. Okay, and if the root is cut longitudinally like this, then you see the inside of the root. This is another image of a longitudinally cut root tip. You see, this is the area of maturation. I think I'm going to start from here, from the tip. This is the tip at which you will find the meristematic tissues. Here all the cells are rapidly proliferating, and here is where the new cells are formed. Okay, and that's why this part consists of, it says, apical meristem, and the outside of the root tip is protected by root cap again. Okay, and then after new cells grow, the new cells will elongate, and here they are elongating in this area. They become long and long and long until it reaches its maximum size, and once they reach their maximum size, they will be differentiated. They will experience the differentiation. Some of them will become the epidermis, some of them will become the root hair, some others will become the parenchyma tissues, some other will become the vascular tissues, xylem and phloem. Now let's talk about anatomy. This is the cross section of monocot root, and you can see the parts of a monocot root like as follow. We have the epidermis, the outermost part of plants, very likely is epidermis. Okay, epidermal tissues. So this is epidermis, and then just inside the epidermis we find the cortex. Okay, and after cortex we can find the endodermis. Endodermis, endo means inside, dermis means layer. Okay, so it's like epidermis, which is located outside, but since it is inside, we call it endodermis. Okay, right after endodermis there are one or two, a couple of layers of cells called pericycle, endodermis and then pericycle. And going deeper, we have these hollow parts. This is actually the xylem. Okay, the xylem. And then in between two xylems we can see this one. Notice that? This one brighter yet smaller. They are the phloem. So just for you to understand, commonly if you look at the cross section of both root and stem, you will notice that xylem are usually larger and they are located deeper, while phloem usually smaller and they are located outer. Okay, again, xylem bigger, deeper, phloem smaller, outer. Hopefully that will help you to remember them. Now you will notice also that deep inside at the center, you can see that these cells are quite similar with these, right? Why do they look similar? Because both this one located in between the epidermis and endodermis, and this one located inside the endodermis are both parenchyma tissues. parenchyma tissues. Okay, they are the same, but since the locations are different, then the parenchyma tissues located in between epidermis and endodermis is called cortex, while the other one, parenchyma tissues located inside the endodermis, is called pith. Okay, cortex, pith. Now this is the cross-section of monocot roots and their parts. Continuing, this is the diagrammatic picture of what I said before: cortex, epidermis, pericycle. Zooming in, we have cortex, so epidermis would be up there, cortex. This is endodermis, and right after endodermis, inside we have the pericycle. Okay, and you will of course notice that at the center of the endodermis there is this part like band, which given darker color. Okay, this is what we call the Casparian strips. Okay, Casparian strips. The Casparian strips are waterproof. Water cannot pass through the Casparian strips. That's why talking about the movement of water from the environment into the xylem, because xylem will bring water and whatever dissolved in it up to the leaves. At first, from the root hair and from the surface of the root, the water will just enter, and they will just enter easily just because the concentration gradient from outside compared to the inside. I mean, the water outside very likely to have higher concentration of water. The water inside very likely has lower concentration compared to outside. Due to that concentration gradient, the water will automatically enter from the environment, from the soil, into the root. So they will enter the root from or through the cells, from cell to cell, or they will go through the intercellular spaces, yeah, like this, yeah, from the side. But once they arrived at the endodermis, because of the presence of Casparian strips, they can no longer pass through the endodermis from the intercellular cells. Okay, intercellular spaces. They need to enter first the cell and then go through these doors called plasmodesmata. Okay, the movement of water through from cell to cell is called symplastic pathway, and the movement of water through the intercellular spaces is called apoplastic pathway. So once the water arrives at this point, endodermis, there will be no more apoplastic pathway. There should be only symplastic pathway. You see here now the if before you see the root of monocot, now you are looking at the root of dicot. The difference between the two, one of the differences is this part. Look at this. Okay, so I want you to let's go back. So I need to also inform you that endodermis and everything inside it is called stele. S-T-E-L-E, stele. Endodermis and everything inside it is called stele. Okay, and in monocot root, in the stele we find parenchyma tissues called pith. But let's take a look at the dicot root. This is the stele, endodermis and everything with it. inside it. I mean, you see there are the large cells, large hollow tracts like this, and there are the smaller ones. Like I said, xylem, larger or bigger, deeper. Xylem, bigger, deeper. Phloem, smaller, outer. And you also can see now that in a dicot roots still there is no pith, there is no parenchyma tissues. Okay, so only epidermis, cortex, endodermis, the blue one, and then the dark line inside this blue part is called the Casparian strips, remember? And the yellow part right after the endodermis is the pericycle. Okay, and after pericycle, so again from the blue circle end, we call it stele, and after the pericycle we can only see xylem, larger or bigger, deeper. Xylem, bigger, deeper. Phloem, smaller, outer. Okay, these are the real image of them under the microscope. Okay, let's continue. And another diagrammatic picture of the difference between the two, dicot root and monocot root. Okay, you can just pause this video and look carefully. Carry on. Now the stem. Here is the dicot stem. Okay, what we need to remember about the dicot stem is that, oh yeah, sorry, the difference between root and stem, when we talk about the vascular system, xylem and phloem, take a look. In root, xylem and phloem are separated. In root, phloem and xylem are separated. They are not in a particular set or bundle. Let's take a look at the stem. In a stem, you see this part, this part, this part, this part, this part. It's like they are what is that? A default set of something, right? Okay, so I want you to take a look at this one. Larger, deeper, larger, deeper, larger, deeper. They must be a xylem. And outside, located more outside, this one, see, larger, deeper. Okay, xylem. Smaller outer, phloem. Smaller outer, phloem. Larger, deeper, xylem. Larger, deeper, xylem. Smaller outer, phloem. Okay, and in dicot stem, there is something in between the two, xylem and phloem. We see this one, this one, this one, this one, this one. We call them cambium. Cambium. Cambium is the part that's responsible in having secondary growth. Secondary growth will enable a stem to grow so big, so wide. Yeah, that's why the dicot plants, including the gymnosperms, they have a very wide, very large stem. For example, in gymnosperms you have redwood tree, right? Sequoia sempervirens. They are so huge. And in dicots you can find, for example, pohon mangga. Okay, mango tree, huge, could be very huge. Okay, so this is dicot stem. Another thing you have to look at in a dicot stem, which will differentiate them from the monocot stem, is that you see this set of parts, xylem, phloem, and cambium, is called vascular bundles. And if you notice the arrangement of vascular bundles in dicot stem, they are arranged like this, concentrically. Yeah, depends on the shape of the stem, of course. See, this is a circle, circular stem, so they are arranged like a circle. But this one, it's like it has four-sided stem. So look at the arrangement of the vascular bundles. It will be adapting to the shape of the stem. Okay, now let's carry on. This is monocots. If in dicots we see that the vascular bundles are arranged like circularly here, they are arranged regularly in a particular arrangement, while here in monocot stem, as you can see, this is the vascular bundle. This is the vascular bundle. The vascular bundles are like scattered. They are not arranged in a particular position or arrangement. Okay, and again I want to remind you that the larger, deeper is the xylem, smaller outer phloem. Larger, deeper xylem, smaller outer phloem. And in monocot there is no cambium. In monocot there is no cambium, means there is no secondary growth in monocot. In this picture you can see the difference, the comparison between the two. What is this on the left, in which the vascular bundles are scattered? Of course, the one on the left is monocot stem, and the one on the right, in which the vascular bundles are arranged in a particular order or position, this is the dicot stem. And as comparison, in dicot stem we can also find pith. The cortex is not really huge, not really big, but it got pith. But here, since there's no endodermis in monocot stem, so we don't differentiate the parenchyma tissues into cortex and pith. Instead, we will just call the part in monocot stem as parenchyma. Okay, the parenchyma tissues or ground tissue. Okay, carry on. From the outside, if we look at a stem, we can see that this is the part of stem from which the branches come out, from which the buds come out, from which the flower come out. Okay, from here there will be no branches, there will be no buds, there will be no flower, and this is another part from which branches will come out, buds and flowers. So this part is called nodes, while this part is internode, a space in between the nodes. Okay, in bahasa Indonesia nodes is buku, internode is ruas. Okay, now the leaf. This is a picture of a leaf, and this picture shows us the parts of a leaf. Blade is actually the sheath of the leaf. Okay, it's like the layer, this large, wide, thin part found in plants, blade. And you can notice that inside the blade we can see the vein, and then the pattern of a vein, how the veins arranged will give us clue of whether it is a dicot or monocot leaf. I mean, what you see here in this picture, the veins are arranged like this. We call it reticulate. Okay, reticulate. This is a dicot. If it is palmate, like a palm, it is also dicot. But if the veins are parallel, then very likely it is a monocot. Okay, you also see this part. It's like if it is peeled, then we have this called waxy coating. Waxy coating is the layer on the surface of the leaves that made of wax, okay, made of wax, and it causes the leaf to have this glossy appearance. Okay, petiole is the stalk of the leaf. This picture shows us the parts of a leaf. You see that the top part is the cuticle or waxy coating. Okay, waxy coating, and notice this part and this part. Like I said before, that the outermost part of plant's body parts of the epidermis. Okay, so we got the epidermis here, we got the epidermis also here. Since it's located at the top, we call it upper epidermis. Since these ones located at the bottom, we call it lower epidermis. Okay, and all parts of plants, root, stem, and also leaves, filled up with kind of tissue we've talked about before, the parenchyme tissues. And same like the other parts, leaves also filled up with parenchyme tissues. But in leaves, the parenchyme tissues got a specific name called mesophyll. Mesophyll. Okay, and you can also see that there are two types of mesophyll. One is like rod shape like this. Yeah, it's like pillars. This is what we call the Palisade Mesophyll, while the other one is like this. They are not too close to each other. They then create hollow spaces here. They look like sponges, so they are called Spongy Mesophyll. Okay, and this is the vein filled with xylem and phloem. Xylem and phloem are arranged in a vascular bundle just like in a stem. Now, look at this part. This is an opening found at the bottom underneath a leaf like this. Okay, this is what we call the stoma. Stoma is a singular form. The plural form is stomata. Okay, through this stoma, there will be gas exchange, oxygen, carbon dioxide, and so on. Okay, how can a stoma open and close? By the activity of these guard cells. The guard cells will determine whether there will be gas exchange or not. Okay, what causes the guard cells open and close? It's about it's the turgidity, the amount of water inside these two cells. Now, this shows us the stomata. I said stomata because I see 1, 2, 3, 4, 5 and a half. Okay, plural stomata, singular stoma. These two look like lips. They are the guard cells. Okay, while the other cells around called the neighbor cells. Okay, let's do a review. What is number one? Number one is, yes, cuticle. Number one is cuticle. What is number two? Upper epidermis. Good. What is number three? Palisade Mesophyll. What is number four? Spongy Mesophyll. What is number five? Lower epidermis. Good. What is number six? The hole, the opening. Stoma. What is number seven? Guard cells. 8. Xylem. 9. Phloem. 10. Xylem and phloem. Vascular bundle. Thank you for paying attention. See you again in the next video. God bless you all.